Okay, if you have a look at your handouts, then we'll sort of try and work with them. I may miss bits out occasionally because I'll have forgotten I've put it in there. It starts off then biologically important molecules, so I'll quickly run through that in a minute. But then you've got a bit on carbohydrates. I'm going to add extra notes to the board. There are also spaces for drawing things in there. So we've got a bit of a space for a glucose molecule down at the bottom. Over the page, a bit more on glucose and the condensation and hydrolysis reactions that we're going to look at with those. There's then a sort of family tree diagram of carbohydrates, which is probably a relatively good starting point, but I'll leave it until later on. Some other diagrams of different types of carbohydrates and some uses of carbohydrates on the following 2 pages before it gets onto lipids. Okay, so you've got 2, 4, 6, 7 sides related to carbohydrates there. The beginning part of it, just the bit before we start the carbohydrates, you've got this idea of, depending on the organism you're looking at, of course it's mostly water, but that percentage varies. So I've said 60 to 95%. I believe we're something around about 65 to 70% water, but obviously, you know, there are organisms that are almost entirely water, aren't there? So things like jellyfish, good example, yeah. So yes, you've got varying amounts of water. We're not concerned with that, strangely enough, but water is an incredibly important component. I mean, we've been meeting it all over the place, haven't we? We've done osmosis, we've done water control through the kidneys and all that, so we'll pass that by on this. But the rest is built up from basic building materials, which are largely composed of carbon, hydrogen and oxygen with a bit of nitrogen. But you're going to need to be familiar with those 4 bullet pointed bits there. Amino acids are the building blocks for proteins. Monosaccharides are the basic units for carbohydrates, but we also find them in nucleic acids. They're simple sugars. Fatty acids and glycerol make up the lipids, which are the fats and the oils. And then we also have aromatic bases, and that's a curious thing that, yeah, we'll leave those till we meet them. But in the nucleic acids, again, that's DNA and its close relation RNA. And you join them together to make what we describe as macromolecules, large molecules. And if you put together many units of the same type, you then have a polymer, and the individual units are monomers. So you've got the examples there that the bonds between the subunits in a macromolecule, I should have put polymers, I suppose, really there. But if you've got the repeating units, yeah, it's a polymer. If you've just made a large molecule, it would be a macromolecule. You make the bonds between the component parts by this process of condensation, removing water, and then you can break them apart, putting the water back in. And we will meet more of that as we go along, so I'm not going to spend any longer on it. I'm just going to get straight into the carbohydrates. Now, the name carbo obviously means carbon. The hydrate bit means what? Water, yeah. So what you have in carbohydrates, you'll see they've got a general formula there, which is written C with a subscript X, and then H2O, as in water, but with a Y. In other words, X and Y don't have to be the same numbers, but you will always have twice as much hydrogen as you do oxygen. So to give you an example, we can have, where X and Y are both 6, we can have C6H12O6. All right, we'll meet that first in glucose. But I'm going to give you another one, C12H22O11 is another one. All right, that's sucrose. And in fact, if you look at it, it's like sticking 2 glucoses together but taking a water out. And that's, you know, that is what happens. That's the condensation process. Okay, now we classify, well, the basic units are the monosaccharides. Mono meaning single. And then the saccharide bit, I think, really means sweet All right, so it's the sweet bit, the sugar bit, so single sugar. And we've got various options here. We classify them according to the number of carbon atoms. Well, that's not a surprise, is it? But this is where biologists don't play the game as far as naming in chemistry is concerned. Because if you look there, you'll see if you've got 3 carbons, now in organic chemistry, we'd say something with 3 carbons started, well, how would its name start? Yeah, well, you got prop. And then, but of course you can give these correct formal chemical names. It's just that if they've got 3 carbons, they're known as trioses. Now, a triose is the first type of sugar that's made in photosynthesis. All right, I'll try and mention photosynthesis a few times this afternoon because photosynthesis is about making carbohydrates, and it'll make Maria's job next week doing photosynthesis a bit easier if you've at least heard the word associated with some of the sugars, because otherwise it's not going to make sense until we get further along. So trioses are the first sort of carbohydrates that are made. You have 4 carbon sugars are less common. Tetroses, we don't really meet any. 5 carbon sugars, pentoses, we're not going to look at them in this section on the carbohydrates, but we will meet them when we do the nucleic acids, because the first part of the name of DNA or RNA relates to the sugar, and that sugar is ribose. In RNA it's ribonucleic acid, and in DNA it's deoxyribonucleic acid, and that ribo bit means ribose. So ribose in the nucleic acids we will be coming back to in that section, but the ones that we meet most often are the hexoses. And I've given you the 3 examples of hexos that you're going to meet. And anybody watch University Challenge last night? By the end of this session you'll be able to answer that question on disaccharides. Yeah, it was such a simple question. We need to rerun it on YouTube or something, don't we, or iPlayer. Yeah, they asked what 3 disaccharides were made of. They asked for the monosaccharides that made them up, and it amazes me sometimes how simplistic some of those questions can be, and you'll realise what I mean when we get to the end of this. That it's not a university standard question. There are a lot more than this, but the classic ones that you're going to meet, they're listed there on your sheet, but glucose, fructose and galactose. Right, and we can combine them together to make other bigger molecules, which is where we're heading with this. And they do have proper names. But, you know, I think if we tried to name them, I think I've showed you the proper name for glucose. I will find it at an appropriate point in a minute. You'll see on your sheet that the glucose molecule is drawn out as a straight line. So you've got the linear representation of it, but in fact that's not how it usually exists. You don't usually find glucose in that form. It's usually arranged in a ring shape, and you will find that on one of your handouts. I've sort of overlapped information here. You will have trouble fitting this into that space unless you can draw really small, but the glucose molecule, if I put all the bits in, which of course we know we don't need to, and this is why we do the skeleton diagrams in organic chemistry. But you have, if I was to put the whole lot in, CH2COOH, I'm not going to have room, I'm sure. Not left myself enough space here. There's bits on the bottom here I don't know, they don't stick in my memory well enough, these. All right, but that would be glucose. Now, we could get the molymod beads out and you could create that, but yeah, you don't need to. That's on, yeah, on the next page you'll find that's the next bit. The yeah, down at the bottom it tells you that the H and the OH on carbon atom 1 can be arranged the other way up. So you could have the OH going upwards and the H going down. And that makes a difference between two isomers of glucose. So if we'd got the OH at the top and the H at the bottom, that would give us beta glucose. Whereas the H up and the OH down, that's alpha glucose. So we already have isomers. And it's amazing, considering the small amount of difference between those two, how much difference there is. If you join them together in chains, if you join alpha glucose together in chains, you get starch. We're perfectly capable of digesting starch, aren't we? If you join beta glucose molecules together, you get cellulose. We can't digest cellulose. Very few things can. Only microorganisms can do it. And it's because of the enzymes and it's because of the shape of the bond. We'll come on to that as we go through. So you've got these two different isomers, different forms, different shapes, but of course their formula is identical. Now, if you turn over the page, you'll see this bit that says standard representation for alpha and beta glucose. And the way it's usually done, I'll just try and, there's a reason for this, and it comes from the exam boards at A level, and particularly AQA, which is the one I've always used or been involved with. And curiously, it doesn't always suit the universities to summarise it like this. But the only important items on that molecule are really the H's and OH's that are sticking off at each end. Now, it's all very well to say sticking off, isn't it? I haven't identified any numbers on here. It's how the heck do we number the carbons when we've got a ring? It is a little harder to do. I don't know if are they numbered on any of these? No, they're not. On your diagrams there, they're not numbered. However, it should say, yep, I'll put them in colour so that we can see. That's carbon 1, 2, 3, 4, 5, and 6. And the only things that are actually of any interest to us, because all we're really going to do at this level is join them together, take them apart, join them together, take them apart, join them together in photosynthesis, take them apart in respiration, digestion. So we're going to really only be interested in the bits that join together, and it's the H and the OH that are attached to carbon 1 and carbon 4. We can ignore all the H's and OH's everywhere else. Now that makes for rather peculiar abbreviations, because what we end up with is something like this. All right, so this is not a standard skeleton diagram, is it? Because I've missed off the OH's, which would be there on a skeleton diagram, that are on carbons 2, 3, yeah, and 6. All right, but that's alpha glucose, the standard abbreviation. Whereas for beta glucose, we just want those the other way up. So same thing. But those the other way round, okay? So you can draw those into those spaces, and if you want to highlight the difference on those, then you can do that, because it's going to make a difference when we try and join them together. The shape of the bond will be different between them. When you say next to each other or diagonal, if they're both at the bottom, not as far as I'm aware. the different isomers have all sorts of different identification sort of apart from alpha and beta, you've got things like R and S and L, and I can't remember the different isomers now. I shall have to go back and learn them if I'm going to teach on this foundation degree. But that's not next year even. So you have to remember with this though that these are not flat either. They are three-dimensional. And you will see diagrams that have the bond shapes in them. And if you look on, you've got a sheet of diagrams that show the shapes of the molecules. Where you get thick lines, those are coming out of the page towards you. They haven't put the dashed lines going away, but it does give you a small idea of the sort of three-dimensional nature of it. But the glucose is often described as existing in two, and I can't think what the description is, but two shapes. It'll either form a sort of shape like that, which is known as the boat conformation, or it forms a shape like that, which is the chair. So, you know, you could sit in here or you could sit on there. But the problem with organic molecules is they tend not to just stay in one shape all the time. You know, you can rotate atoms around bonds, and so they do take up these different conformations. It's a good description of them. All right, so here's our alpha, here's our beta. Those are the abbreviated forms of them. The other monosaccharides are on the sheet. Well, they're all on this sheet, okay, which is the other side of that one. So you've got, across the top there, you've got the three hexoses, the glucose, fructose and galactose. Now, if you look, glucose and galactose are very similar in terms of their layout. They're both hexagons. But you've got a difference on carbon 1 and carbon 4 with galactose. So it's curious, isn't it? There's very little difference between them, but it makes it a completely different sugar. And in fact, you know, galactose and the beta glucose are even more similar, but you've only got to switch those around. You've got a different sugar. You've got galactose. Fructose slightly different because it forms a pentagon, and you've got two carbon branches coming away on, you know, one on each side there. And then you've got ribose and deoxyribose underneath that for the 5 carbon sugars. And I'm just trying to find out, oh yeah, it's bottom right hand corner is where you get the deoxy bit, where you've got a carbon that's just got 2 hydrogens attached to it rather than a hydrogen going up and an OH going down. So like spot the difference game, that one. So those are the monosaccharides. They're repeated again underneath because that was just part of an image where I actually wanted the disaccharides and the polysaccharides, but you got the monosaccharides repeated again for the hexoses anyway. And what we need to look at now then is how we're actually going to join monosaccharides together to make a disaccharide. So you've got some space. We're going to do some drawing in those spaces, but you've got 3 common disaccharides there, and that is literally the answer to last night's University Challenge question. And they went through them, maltose, lactose, sucrose. It was even in that order that they did them, I think, and said, what are the monosaccharides that make up these disaccharides? Disaccharide is obviously 2 sugars, and we're going to join them together by the process of condensation, taking out a water molecule. Well, you look at this, you say, but there isn't a water molecule there. No, but there's enough H's and O's to make plenty of water. So what we're going to do is take away 2 hydrogens and an oxygen from between the 2 molecules and join them together that way. And I shall probably have to check that I take them off the right place. It's funny, it's one thing I just haven't committed to memory for some reason. So you've got, if you join glucose and glucose together, you get a sugar called maltose. Where do you find maltose? Maltesers, yes. Yeah, people always say that thinking that that's not going to be right. but yeah, malt, basically. Beer, yeah. Well, you won't in beer actually have very much maltose left, but beer comes from malt. Yeah. So what is malt? That's often the tricky question. Maltose is a sugar that's in malt. And it's got a distinctive taste, hasn't it? You know, Maltesers, malted milk drinks, that sort of thing, malted this, that and the other. And malt has, it's got a, I don't know, not caramelly, but a different sort of flavour. It's got quite a distinctive flavour, hasn't it? So what is malt? Well, malt is germinating barley grains. All right, now that is where beer comes from, isn't it? Beer is made from malt. So you get the malt, you get the barley grains. They're soaked, I think, to start with, and then they are left to germinate. And that is the first stage of it. You get your germinating barley grains, because what they're doing, barley grains don't store maltose, the sugar. What do grains usually store? Do you think? You know, think of bread and stuff like that, and rice, starch, yeah. Now starch is big long chains of glucose. Let's say it's 1,000 glucose units joined together. Maltose, the sugar, is just 2 glucoses at a time. So what happens when the barley grains germinate is that the starch is chopped up into maltose so that the grain has got sugars that it can use to grow. So a germinating grain takes its stored starch, breaks it down into maltose, and then uses the sugar for energy in order to grow. So you've got maltose in malt. And then after that, of course, you know, you can ferment the sugar rather than the barley grain using it. You can get yeast in on the act, and the yeast will use the maltose and it will ferment it into alcohol. All right, so maltose, yes, it is in Maltesers and in beer, and anything that says malt has got that sort of sugar in it. Okay, the next one down, lactose. Glucose and galactose joined together into a disaccharide. Where do we find lactose? In milk, yeah, and any sort of milk-based product, so whether that's cheese or yogurt or anything like that will have lactose in. And of course, in order to actually use lactose as a food, we would need to be able to digest it, to break it down back into its glucose and galactose, but not everyone can do that, can they? So some people are, that's it, lactose intolerant. They are normal. It's the rest of us that can digest lactose that are freaky weirdos. Okay. Technical term there. But if you think about it, as adults, should we be able to digest milk? It's actually a little bit weird, isn't it? Because milk is intended for babies. Yeah, baby mammals drink milk. So adults using milk is actually a little bit strange, isn't it? But of course, it sort of goes nicely with the end of it's the end of the next unit, actually. We do a bit on evolution. And of course, it provides a survival advantage, doesn't it, to be able to digest something else. And so what would have happened in the past is that some individuals would have had a mutation that meant that they could digest milk. And of course, they would survive in times when there wasn't much food, because they could get milk from animals and live on that. And it's a good source of nutrients, isn't it? So, you know, it would give a big survival advantage. But yet you do actually have to wonder, the first person who milked a cow and drank it, what did they think they were doing? You know, anything. It's, yeah, you've got to think it must have been that sort of feeling. Well, you know, that's it. But I mean, you know, they've seen other animals. Well, they're not other animals. They've seen baby cows, calves.
Surely the first person who milked a cow would have known about breast milk.
Well, they would know about human milk, yes. So they, well, yes, yeah. It wouldn't. No, I know it's not a huge step, but no. So but that's so. There are some quite just sort of odd connotations, aren't there? Yeah, I mean, why wouldn't you know, perhaps have tried eating the grass? Well, perhaps they had tried eating the grass, but that wasn't so easy to digest. But you can imagine if you've got, you know, you go back into our history and you've got the first humans sort of starting to settle and starting to develop agriculture perhaps, and perhaps having herds, well, you know, keeping a few sheep or goats or something like that. But if you can move about, if you're looking after the animals and you can move about, let's say, to better pastures, up hills and what have you, and you aren't running short of food because you can actually use the milk from those animals, your whole little community is going to benefit, isn't it? So you can see that being able to digest milk is actually it's a evolutionary advantage. But of course, those who can't, as adults, will usually start life being able to digest milk as babies, and then what they do is, well, what we do is produce an enzyme called lactase. ASE means an enzyme, OSE is a sugar. So lactase digests lactose. And normally you would think, right, when a baby mammal has finished getting milk from its mother, it will stop making lactase because it's a waste of effort if you're not getting milk anymore. And so there is a sort of natural switch in the body, and we'll look at the control of gene expression in the next unit, that would normally switch it off. And in some people it does, and that is essentially normal, isn't it? But for the rest of us, we don't now have that switch. We don't switch off our lactase production, so we can keep digesting milk. But not everybody can. There is one part of the world particularly that tends to be lactose intolerant. China. Yeah, you don't find dairy in Chinese food, do you? No, there's no dairy in China. Oh, yes, and yak milk and all sorts. So yeah, it's interesting. Yeah, China particularly. Yeah, I mean surrounding countries use a lot of dairy, don't they? Ah, right. Well, that would figure, wouldn't it? That you know we will have some sort of smell of digested dairy products, perhaps. So yeah, I probably shouldn't record this bit, but you know, you've always got this option that you know if you want to attack China, you know, just drop a lot of milk on them. We had the idea of cheese bullets and yeah, yogurt bombs and things like that. Sort of yeah. But the problem that you have if you can't digest lactose is that the bacteria in the gut will start to ferment it and produce gas. It also drops the water potential in the gut, so that pulls water out of the body, so it makes the contents of the gut very watery. So you get that wonderful combination of gas and diarrhea, you know, which is not good. And of course that comes with a lot of pain as well. Some people it's more serious than others, but there are other animals as well. I mean, cats are largely lactose intolerant. Yeah, cats are. A lot of cats can tolerate ordinary milk, sort of cow's milk, but you do get the bottles of milk, especially for cats. I hesitate to say cat's milk because it's not. It's cow's milk. Yeah, milking cats is not an easy job. Yeah. But they take cow's milk and they treat it with lactase enzyme, and it breaks down the lactose. And of course, you can get that for people who are lactose intolerant. You get lacto-free, don't you? And it's just it's ordinary milk, I should say, cow's milk that's been part digested, if you like. It's had the lactose taken out of it. But yeah, giving cats cow's milk, or I mean, a lot of them like cream and cheese and stuff like that, can be a bit risky. Long life milk is just ultra heat treated. That's what UHT means. So it's well and truly sterilised, so it lasts longer. That's all. It's got all the normal things you'd find in fresh cow's milk, but it gets changed slightly. It's just cooked a bit. That's all. So that's the maltose and the lactose. Sucrose, the other one, probably the most common one, isn't it? Because that's just, you know, household sugar, sucrose. So glucose and fructose joined together will give you sucrose, and that's the one that's most commonly in food and, you know, the one in the sugar bowl, that sort of thing. So what we need to look at then is how we're going to join together two alpha glucose units we're going to start with, and we're going to create maltose from those on this diagram. And I will have to check because for some, oh, no, hang on, let me start by drawing them in. We just need the abbreviated version, all right? So we'll have our, get these the right way up. Whoops, so there's one. So 2 alpha glucose units side by side, like that. Now, if you look at the area where the join is going to occur, fairly obviously in this sort of area between the two, then you can see plenty of material available for the formation of a water molecule. You've got 4 hydrogens, you've got 2 oxygens, so you've got plenty to choose from. But this is where I will just need to stop and check. So I'll just put this on pause. Okay, what we're going to do is remove enough atoms to make water by removing those. All right, it's not the most obvious choice necessarily. You know, you've got those loose hydrogens up at the top there. You think, well, couldn't you take those off? Well, no, because they're actually more strongly joined to the carbon than the H here is joined to its oxygen. If you now think what this means in terms of bonding, what we get left is the bond between the oxygen and hydrogen. Let's use another colour just to show what's going on here. Here, all right, is a pair of electrons. The bond between the carbon and the oxygen here is a pair of electrons. What we're doing is taking away one of those electrons and leaving an unpaired one. So what we can then do, I've now made a bit of a mess of that. Doesn't really matter. What we can now do is join together the electron that's left over here with the electron that's left here, and we can bond this carbon to that oxygen. Right, that's a real mess. All right, but it just means looking back at your bond formation stuff in the chemistry unit. All right, so let's start that again. There's my OH. Whoops, so there's my other OH. And so that is what we're going to remove. And so where it says minus H2O, that's the condensation process. So you can see it's not water that's being removed, it's a hydrogen off one side, off the carbon 1 over here. It's the OH group off the carbon 4 on that side. This is the process that's called condensation. And what we then get left with is the oxygen from the carbon 1 here, joined to carbon 4 on the other side. So we've now got to redraw these things again, but join them together. So we have all right, what we've got left here is that, okay, and that oxygen there has got an odd electron that it was sharing with the hydrogen. The hydrogen's been taken away, so we're going to share now with the carbon 4 on, sorry, that got a bit squashed so that I could fit it in, and you get a rather untidy maltose looking like that. It's one of the hardest things to do on the board is draw regular shapes sort of repetitively. So these are our monosaccharides, which are already labelled on your diagram. Here are your disaccharides, well, a disaccharide. And of course, having put them together, now we've made maltose. We could break that maltose molecule apart at the same point by putting water into this bond, really, is you know what we're saying. We're going to break that bond using water. But it's not just water. You've actually got to break a water molecule apart and put an H on one side and an OH on the other. But what that means ultimately is that we are adding water, hence we call it hydrolysis. But as I said, it's a word with two meanings. It can mean splitting the maltose molecule using water, or it can mean splitting the water itself. Water molecules can be split in various ways, and that happens naturally to some extent, but it also happens when you dissolve things in water. But it also happens in photosynthesis. Thought I'd just throw that in. One of the first things that happens in photosynthesis, obviously you know photosynthesis requires light. One of the things that light does is to split water molecules apart. It's called the photolysis of water. So the photo bit means light, and the lysis again means splitting. So photolysis of water splits the water into hydrogen and OH for the other half. So H plus and OH minus, if you like. And the hydrogen is then used to turn carbon into sugar. So we've got not polymerization exactly, I suppose we've got dimerization. So these are our monomers. This is not a polymer. Poly means many. It's a disaccharide, so it's a dimer. If you added another one on, you'd have a trimer. After that, yeah, well, you know, the chains get longer and longer. Where it becomes a polymer, I'm not exactly sure. You can apply the same sort of principle to the other sugars as well, but it's just not quite so easy to join them together. But that is, just to complete it really, is where the the two H's and the O are coming from and going to. And so in the formation of a disaccharide, we form a new bond. Now that new bond's got a name, not just any name. It's called a glycosidic bond, which you can see at the bottom there. Now, where is the glycosidic bond? We used to, and I used to mark it right on exam papers, and yeah, I'm not entirely sure, but just this whole thing here used to refer to as a glycosidic bond. But actually it's not, because this is the original bond, isn't it? So actually, if we're going to be precise, it is that, but it is sort of the whole join, if you like. But this idea of a glycosidic bond is between any sugar molecules that are joining together. Doesn't matter how many of them. And the next step we've got to consider is, what if we want to join together lots of them? Now, you have this this sort of family tree diagram of carbohydrates, and it sort of just gives you an overview of how we classify them and some of the characteristics of them, their properties, if you like. And you can see that we've got sugars. We've now dealt with sugars, really. We've done the monosaccharides and the disaccharides. But characteristics of sugars, all right, over this side on here, you can see they're small molecules, so relatively low formula mass. They're sweet. It's one of the things we associate with sugar, isn't it? The fact that it's sweet. But they're not all equally sweet. Come back to that in a minute. They're readily soluble in water, they're crystalline, and the suffix, that means at the end of the word, "ose," is used in naming them. All right, so sugars will always end in "ose." This idea about them being sweet, there are various sort of scales that are used to measure sweetness. One that I've come across that is quite a useful one to keep in mind, really, is one where sucrose, just our ordinary domestic sugar, is just given an arbitrary sweetness value of 100. But that's composed of course, sucrose is composed of glucose and fructose. Yeah. Now, if sucrose has a sweetness of 100, compared against that, glucose actually only has a sweetness of 80, but fructose has a sweetness of 120. Fructose is more sweet than glucose. Now, the theory sort of then that goes with this, it's something that we have to say over and over again in GCSE biology, so I don't know if James can remember the fructose thing. Vaguely, slimming foods. Yeah, what they say in GCSE is that fructose is very useful in slimming foods because you can use less of it to get the same sweetness. But in fact, fructose is quite expensive, so that's very often not done. And there is an idea that fructose actually has quite a severe effect on blood sugar levels generally, and high fructose products are generally thought of as being a bad thing in food. So it's it's got its uses, but this idea of sweetness is an interesting thing. Obviously, we know lactose is a sugar, but we don't think of that as being sweet, do we? So presumably, all right, glucose on that scale was 80. Presumably galactose is much less than that, perhaps only about 20. So, it might average out at whatever's in the middle of those two, about 50, 60, something like that. On the other hand, you've got other products that are sweet, which are not sugars. It's not only sugars that are sweet, is it? You've got the artificial sweeteners, haven't you? All these, somewhat dubious, perhaps, chemicals. So what's the most dubious one? Aspartame? Yeah. There's a whole big conspiracy theory around aspartame that people say how awful aspartame is. On this scale, it's got a sweetness that goes into thousands, so you don't need very much of it. Okay. Now, aspartame is actually made from 2 amino acids. The reason people think of it as being an awful, poisonous, bad thing is that when it's digested in the body and processed by the liver, it produces an alcohol, and that's methanol. Not ethanol, which we're familiar with, but methanol, meths, if you like. All right? And meths is phenomenally toxic. But how much sweetener do you think you would have to use to produce a toxic level of methanol? Because we don't use much at all, do we? Because it's so sweet. There's probably more methanol coming from other products in our lives than there is from metabolizing aspartame. But that, as far as I understand it, is the main argument against it. It's actually made just from 2 amino acids that you'd find in, the most common sweetener, the aspartame one. There's others as well, aren't there? There's sorbitol. Now, sorbitol and other ols, those are alcohol-based. They're sort of alcohol sugars. Sorbitol, mannitol, xylitol. Yeah, there's a whole bunch of ols which don't raise blood sugar levels directly, but they do have other interesting effects. They are, yes. They're useful in things like diabetic sweet stuff. But there's always a warning, and if you think, sugar-free this, sugar-free that, then you have to look carefully because it does have a laxative effect. I found that out the hard way once. It was, because trying to cut down sugars is all very well, and you think you found something to replace it with, but eat the same quantity? No, not wise. And then there's one of the, I can't remember which one it is, that contains the amino acid phenylalanine, or maybe that is aspartame. Anybody got a diet drink on them? No? Oh, water or full sugar. If you look on diet drinks, if you know anybody who has diet drinks, you will usually see contains a source of phenylalanine, and that's in the sweetener. And the reason is that the processing of phenylalanine is, phenylalanine can't be processed by people with a genetic condition called phenylketonuria, and it can damage the nervous system. But we'll come back to that another time. But yeah, so sweetness, it's a characteristic of sugars, but not only sugars, all right? There are other sweet substances. And it's more about quantity, isn't it, than anything else? Okay, so what does that leave us with? Well, and you can see these little black circles on this sheet representing our monosaccharides. So a single one is a monosaccharide. Join them together with a glycosidic bond, you've got a disaccharide. And then over on the right-hand side, you've got these polysaccharides. So these are macromolecules with high, I always want to say molecular masses, but formula mass is easier, better. They're not sweet. It does make you wonder why, if they're made of sugars, why are they not sweet? It's, you know, it's just the way they're detected on the tongue. They tend to be insoluble or only very slightly soluble in water, but that's why they're useful. That insolubility is actually the whole point, because they can be used for structural uses, they can be stored, because if they're not soluble, you can store them. They're not crystalline, and we make them by joining together many monosaccharides. So you can see there you've got a lot of little black blobs joined together. So well, let's have a look at these polysaccharides then, and try and distinguish between them a bit. And then we'll have a look at some of the examples that I've got in here as well. The ones that we're most familiar with, starch, cellulose and glycogen. Well, starch and cellulose are both plant polysaccharides. So once a plant has gone through photosynthesis and it's made glucose from, well, it starts with the trioses, remember? It will then make glucose from that. It can then join glucose units together to make starch if it wants to store it, and to make cellulose if it wants cell walls. All right, so yes, if we move over to after the diagrams there, we've then got a little bit on starch, cellulose and glycogen. Starch itself is actually not just one substance, and different starches will have different proportions of the two component compounds in there. They're both created from glucose, but one's known as amylose, the other one's known as amylopectin. So you've got a description there. Amylose is straight chains of alpha glucose, and amylopectin is branching chains, similarly, of alpha glucose. And so what you have, the amylose, what tends to happen with amylose, although it says straight chains, those straight chains tend to form, right, I'm going to have to draw it first, what we'd call a helix. Now, that's actually not very good, but all right, if we join them together along there, you get a better idea. I've done rather more of it than I needed to. I haven't yet mastered the drawing of a helix that goes in, that twists in one particular direction. So you probably realise you can have left-handed ones and right-handed ones, can't you? Do you remember they found a snail not so long ago that had its shell curling in the opposite direction? Remember that? And they were trying to find a mate for it, and they couldn't because, you know, they have to coil, the shells have to coil the same way in order for them to be able to get close enough to mate. So this one snail with the shell that had the helix in the other direction, and I can't remember what they called him, but it was in the news. So what you have is known either as an alpha helix or a beta helix. You very rarely hear of a beta helix, but I think, and curiously, helix, I think, is the Latin name for snail. But this is supposed to be an alpha helix, which I think is the right-handed one. We'll meet them again. We'll meet the helix when we're doing proteins. But it forms this alpha helix. Okay, and that's a representation of amylose. The other one, on the other hand, amylopectin, it forms, it's almost like Oh, I don't know quite how I'm going to do this. It sort of curves around and it branches a lot, and you get this sort of effect. Yeah, now I've got to draw some on here, haven't I? Oh, really ought to have them joining somewhere sensible. It's easier said than done. All right, but that's more what amylopectin or amylopectin looks like. It's not very evenly done, but these branching sort of coiled, it's not going to help actually, arrangement of glucose units on there. So this is the branched. I don't know if there's room to draw both of those in there. It depends how small you draw. But you've got a mixture, right, of each of both of those in starch. And the important thing about starch is that it's not soluble. You know, you can't just break these glucoses away from each other to interact with water molecules. Normally, you see, glucose will interact very nicely with water because, and sugars generally will, because they've got all these OH groups on them, which of course are polar. And water molecules being polar, you know, they can interact round a single glucose or round a single sucrose molecule even. But you can't get enough water to get around this to actually make this dissolve. So what you tend to get with starch is more of a suspension, we would describe it as. Now, if you think about what you get when you've boiled pasta or rice, that you get starchy water, don't you? And that's the starch. It's not dissolved in the water, but it's suspended. If you were to leave it, you can try it, you know, leave some water hanging around in the kitchen for a while, the starch will probably settle out. You might have to leave it quite a long time because we're talking about very, very small particles. But what you start with is a starch suspension. So the water will sort of get around it, but not into it to dissolve it. So starch then, that's alpha glucose. Now, cellulose. Oh, this gets to be fun. I hate drawing this. And we haven't got a drawing. I've got glycogen on that sheet, which saves the one further down. But cellulose. Yeah. Right, with cellulose, we've got a bit of a problem because what we've got to do is join together beta glucose units. Now, with beta glucose, on the carbon 1, you've got the OH going up and the H going down. So what we end up with, if I draw the first one, I'm going to have to miss some of the detail out on this, but here is our first one. Now, because the OH goes up, and that's where the bond forms, it goes up instead of down. But of course, the carbon 4 has still got the H up and the OH down. So what you end up with is the molecule sort of flips over. And I now have to try and draw this the other way up. So that's not going to be right, is it? I put that in the wrong place. All right. Something like that. That's it. And then it goes down. I can do this one. That's easy. And then it goes up again. I should just be able to copy these after a while, but it's amazingly tricky to do. And then it would be going down again. I think I've done that right. Up, down, up. It would be down, yeah. So the idea with the problem that you have with the beta glucose is that the glycosidic bonds are now not the same shape. You know, the whole area of that bond is now not the same shape. So you need a very specific enzyme to break that down. We have met the idea of stereospecificity several times with proteins that we've met. We've met the receptors, we've met, all sorts, haven't we? Channels in membranes and that sort of thing. Enzymes speed up chemical reactions by attaching themselves to, well, specifically to the target area that's going to be reacting. And so if we want to attach an enzyme to break down starch, for example, we have an enzyme called amylase that attaches to amylose and breaks down the glycosidic bonds there between the two alpha units. But amylose, all right, made from alpha glucose, is not going to give us the same shape as the cellulose. So amylase, that enzyme, won't break down cellulose. You need cellulase. So the enzyme cellulase will actually fit the glycosidic bond between two beta glucose units, and very few living organisms make cellulase. So what sort of organisms can digest cellulose? Sorry? Cow, sorry, cow. Yeah, sorry, no, that's me, just not, yeah, cow. Yes, sort of. Any animals that you would call a herbivore, yeah? So, yeah, cows, sheep, horse, rabbit, yeah? They actually can't. They themselves can't do it. They don't produce cellulase, the enzyme. Bacteria, yes, they have bacteria in their digestive systems that do it. So a cow has 4 stomachs, yeah? Heard of that, haven't you? And these 4 stomachs are required in order to digest the cellulose. And that type of animal is known as a ruminant animal. And they use the first 2 stomachs. They have bacteria and other microorganisms in there that will ferment the plant material into something sloppy. And then it comes back up again, and they chew it again, and then they swallow it a second time, and it goes through their normal stomach. But yes, chewing the cud is that thing. It does, doesn't it? Ruminants are grazing, yes. Right. Yeah, I'm not, the way animals are classified, in terms of things like meat and keeping animals and stuff like that, yeah. No, well, rabbits aren't. Rabbits aren't ruminants. Yeah, no, well, all ruminants are grazing animals, but not all grazing animals are ruminants. Other grazing animals, animals that eat grass, have another system for digesting their food. Do you know what rabbits do at night? They do eat their poo. All right, but it's only partly digested. It's not sort of complete waste at that point. What they do, yes, they sleep sort of curled round, so their mouth is as close to the other end as possible. And what they produce is sort of light-coloured, soft material that just is recycled. So essentially, it goes through their digestion twice. What they have, rather than having 4 stomachs to get the cellulase organisms active, they have a very large appendix area. It's not just the appendix, but the appendix and the cecum, which is the bit that it's attached to. And that, yeah, I mean, that particular way of digesting is just peculiar to rabbits, I think, because other animals that have the same sort of digestive system don't do that. Like horses, for example, don't do that, but they're not ruminants. So animals that digest cellulose have a real job on their hands and very often have to use bacteria to do the job. They can't do it themselves. So yes, that's my argument against being vegetarian. You see, we don't have 4 stomachs, we don't have a large cecum and appendix, so we can't digest cellulose. So, you know, and I love the taste of bacon, yeah. That's I was trying to avoid saying it, but yeah, that's what it comes down to. But yeah, I mean, the whole point of vegetable matter, plant material in our diets is actually that we can't digest it. It's fibre. Well, fibre is cellulose. Yeah. So what we have to do is we grind our food up, and when we digest plant material, you know, you eat an apple, you have to chew it up. Well, the nutrients we get are from the inside of the cells, but we can't digest the cellulose in the cell wall. So that will go through undigested. And it's the fact that it's undigested that gives the digestive system muscle something to work on So, you know, we don't get constipated, and the main function of fibre in the human diet is to prevent large bowel cancer. Yeah, yeah, there is a really strong link between fibre in the diet, or lack of fibre, and increased risk of bowel cancer, because it moves stuff through. So if you don't move waste products through the intestine, they will sit there and irritate the lining, so that, yeah, it increases the risk. So yeah, slightly off the subject, but you know, it's all sort of part of it, isn't it? But the purpose, I mean, cellulose for plants, of course, you know, so they use photosynthesis to make trioses, then they'll make glucose, and then some of that glucose will be turned, well, some of it will be alpha glucose for immediate use for energy, some of it will be stored as starch, but some of it will be converted into beta glucose and then turned into long strands, which are insoluble as well. When you get cellulose laid down in plant cell walls, you get layers that sort of go at right angles to each other. It doesn't work when you start adding layers, but layers, so it's layered at right angles in cell walls. Okay, so we've got this very resistant fibrous material that can then be, it's waterproof and it's hard to digest. And of course we do use it. It's the main component of certain things that we make, like, for example, paper is largely cellulose, cotton is largely cellulose. If they were soluble, then yeah, it wouldn't be so useful, would they? Really? Yeah, put your cotton clothes in the wash, and yeah, that's that. So yeah, you know, if you relate structure to function here as we go through, it's quite important. We've got some other examples we'll look at in a bit. So we've had starch for storage, we've had cellulose for structural uses, and then of course we've got glycogen. Actually, I realise I don't need to draw that one because there is a diagram of it on the diagram sheet that you've got directly opposite it. Glycogen, it's like amylopectin. I think it's actually got more branches. And the difference, I don't know really, you know, why they are actually different substances. You know, it's a fine distinction, really. But it's what animals form in liver particularly, but in muscles as well, just for storing glucose. So it's not soluble, but it's there to be used. And of course, we know that, you know, the conversion between glucose and glycogen is under control of the hormones insulin and glucagon, as it says there. Okay, now if we want to test whether something contains carbohydrates, there are several chemical tests. We're going to do these, I think, next Monday. We're going to do 2 lots of practical work on this. You might have come across this. They sometimes describe them as food tests, but they are biochemical analysis, really. And it depends on the type of sugar that you've got there. Starch, we know how you can test for starch with iodine. That's easy. Now, you're actually going to meet that in chemistry as well, using iodine and starch together. So it's funny, everything comes together sometimes. But you can see the other 2 tests there. It's talking about reducing sugars and non-reducing sugars. Glucose and the other monosaccharides are all reducing sugars. They can reduce copper ions from copper 2+ to copper 1+. And the thing about copper 2+ is it's blue, and copper 1+ is red. So if you heat a reducing sugar with copper 2+ that's blue, it will go red. All right, it takes a bit of a roundabout route to that, but it's a test called Benedict's test. It uses copper sulfate, the bright blue stuff we've met a couple of times. And if you heat your solution up with Benedict's, then or your food solution up, If it's got a reducing sugar in it, it will change the colour of it. Non-reducing sugars don't do that, but they are often made of reducing sugars. So you can, if you start with a non-reducing sugar that doesn't change the colour, and then you actually split it apart by boiling it with acid, you can then get it to do that test. But I will explain more about that when we get to the practical bits. All right, so you've got the biochemical tests for carbohydrates at the bottom of that page that we will need to come back to, as I say, next Monday. We're going to do that. We're going to do the biochemical testing. We're also going to do some chromatography, very simple chromatography. Anyone done chromatography before? Yeah? With paper?
I did with the instrument.
Oh, right. Okay. Yeah? Sorry? You can, yeah, oh, you can do it with, yeah, the sweets, Skittles. You can put them on a piece of paper, drop water on it, and the colours spread out. Yeah, and you can, yeah, there are all sorts of pieces of equipment for doing it these days, and you can use glass tubes full of beads to do it. We're just going to do a very simple version with a piece of paper. But chromatography is quite an important analytical technique. And if you're doing anything biological at university, then you will come across chromatography in all sorts of different forms. Yeah, we only really have time, and I only have the expertise to do one. Right, that leaves us with a couple of pages of rather a lot of writing on tables. So I'm just going to pick through these, and then I'm not going to go on and sort of go into the next section yet, because I'm going to leave that for tomorrow. But you've got table 5.6 and 5.7. 5.6, the chief functions of monosaccharides, and then 5.7, further examples of polysaccharides and closely related compounds. You can see that there aren't any disaccharide uses here, because they tend not to exist for very long. They're either built on the way to making a polysaccharide from monosaccharides, or they are produced partway in the breakdown process. So, yeah. With the monosaccharides then, we've got chief functions of, and then you've got quite specific trioses, tetroses, pentoses, hexoses. Quickly going through that, you can see with the trioses, glyceraldehyde and dihydroxyacetone. Oh, those are very, very old names. You know, as far as a chemist's concerned, those names would not really be suitable. However, biologists don't care. And I've got my biology hat on this afternoon, even though it's supposed to be chemistry. So, yeah, as far as biological chemicals are concerned, you know, you can just see the biologists shrugging their shoulders and saying, so what? You know, I'm sticking with the old name, but I might change part of a name here or there if I feel like it. But you can see underneath that, and the reason I wanted to mention this, intermediate in respiration, photosynthesis, and other branches of carbohydrate metabolism, which is not surprising. You know, this 3-carbon sugar, if you were building a 6-carbon sugar, then it makes sense to build a 3-carbon sugar to start with and perhaps stick them together somehow. And if you were breaking down a 6-carbon sugar, then the first thing that happens, and this is taking you right back to the cells unit when you did mitochondria and talked about what happened in the cytoplasm and in mitochondria. And I don't know if you remember, glycolysis in the cytoplasm breaks down glucose into pyruvate. Well, pyruvate is not actually a sugar, but it is a 3-carbon compound, and on the way to pyruvate, you go through a triose. So yeah, just sort of getting back to that. We will do we do respiration at the end of this section, but that's a few weeks away yet. The tetroses, as I said when we put those into the notes there, they're rare in nature, occurring mainly in bacteria. Not discussed further in this chapter. Well, yes, that shows where that came from. The pentoses, a lot of appearance in the nucleic acids, the ribose and ribulose they've got there as well. But they're elsewhere as well. So you've got the nucleic acids, but they also turn up in, and it says some coenzymes, NAD, NADP, coenzyme A, FAD. All of those you'll have met in the next few weeks. FMN, I have no idea what that is. NAD stands for nicotinamide adenine dinucleotide. NADP is nicotinamide adenine dinucleotide phosphate. Right, that's all that was. But NAD and FAD, you can see they're very, very important in respiration. And they actually originate from B vitamins. The reason you need B vitamins is to make those substances which are involved in respiration, which is the release of energy from your food. So if you're feeling a bit tired and lacking in energy, taking B vitamins can improve your respiration and energy release. There are quite a lot of them, yeah, but there are some sort of quite specific ones that are useful. NADP is the version of NAD that you find in photosynthesis. So the NADP is doing the same job. It actually carries hydrogen ions, really. You've then got synthesis of AMP, ADP, and ATP, which are adenosine monophosphate, diphosphate, and triphosphate, yeah. The adenosine, the O's bit in adenosine, right, the O's means sugar, yeah, and it is ribose. You start to see some of these, you start to realise where the words work, don't you? It's, it's one of the things about science language. If you can get hold of those little bits, it's amazing how much you can work out from words you've never seen before. All right, so now you know O's means sugar, then adenosine. Adenosine is adenine, ribose. That'll do. What else we got? Making polysaccharides called pentasans. No idea what they are. And ribulose bisphosphate in photosynthesis. Ah, yes, RuBP. RuBP it's described as. So, yeah, there will be a fair overlap between this and photosynthesis. So it should give you a little bit of a start. The hexose, as we know, source of energy when they're oxidized in respiration. We know about glucose being used for respiration, particularly by the nervous system, the brain particularly. All right, has to have glucose, can't work with anything else. And then, making disaccharides and other larger molecules. Oh, I see there a useful word. You don't just jump straight from disaccharides to polysaccharides. A combination of 2 to 10 monosaccharides. I'd have thought that was actually 3 to 10 because 2 is a disaccharide. Anything more than that, I say 3 to 10, is oligosaccharides. I'd forgotten oligo. So you can have mono is 1, di is 2. You could have tri for 3, but you can go into oligo up to 10, and then poly for lots. You then have monosaccharides can be used to create all sorts of other stuff. So derivatives of monosaccharides you've got down there. Things like you can turn them into sugar alcohols like glycerol. We're going to need glycerol tomorrow morning when we put lipids together. And sugar alcohols we mentioned earlier as sweeteners. For example, mannitol in fucus. Fucus is seaweed, I think, and some fruits. You can also turn sugars into sugar acids. You can turn them into deoxy sugars. You can turn them into amino sugars. There are, a lot of uses for sugars. We tend to think, the most obvious use, glucose, it's a fuel, we use it for energy. But actually, sugars can be used for a lot of other biochemical pathways. Right, very quick look through the other table, and then we'll call it a day. So we had quite a busy one with new stuff. We've got, for the polysaccharides, structure Agricultural uses. Cellulose we know about, so this is other polysaccharides. It says further examples. One of the most important ones, pectin or pectins. If you stick together galactose and galacturonic acid, which is just an acid version of it, you get something called pectin. Now, pectin, it sort of glues plant cells together, you know, because otherwise they would just be a heap of cells. But it sort of holds the structure together in plants because they haven't got a skeleton or anything like that for support. They actually need, you know, to have cells actually fairly solidly stuck to each other. And pectin is really important. Jam, yeah. Fruits that don't contain much pectin are very difficult to make jam out of because the pectin is what gives it the gel. And strawberries are notorious for that. Strawberries can be really wet and a bit short of pectin. So you can buy bottles of pectin called Certo, and you just add that to the jam. Not that I've ever made jam in my life, but you can add pectin to actually help it gel. Other fruits contain a lot more. So something like, say, black currants, I think, contain an awful lot of pectin. They will set as solid as anything without any help. Pectin's also a problem if you make wine, because obviously wine, if you're fermenting fruit, you can end up with cloudiness in your wine as a result. Well done for the last 5 minutes. We're just rambling around some uses of carbohydrates. I'm going to stop in about 5 minutes anyway. But yeah, if you get pectin haze in wine, you can add an enzyme called pectinase to break the pectin down. So pectins are from cell walls, from sticking fruit cells together, of plant cells together. Arrowroot, yeah, arrowroot, I think, is actually mostly starch. Isn't it a very fine starch? Sort of along the lines of corn flour and that sort of thing. So yeah, I mean, a lot of the polysaccharides will tend to form gels because you can make a gel with starch. That's, oh no, I'm thinking cellulose. I'm trying to think what wallpaper paste is made of. So, rambling off the subject. Yeah, don't eat wallpaper paste. It's not good. It's the additives like the fungicides and things they put in wallpaper paste that means it's not a good source of food. You've got to be desperate. So what else have you got? Bacterial cell walls. We spoke about murine when we were doing the staining the bacterial cells, the gram-positive, gram-negative thing. Outer coats of animal cells, you get glycoproteins and glycolipids, and it says over on the right-hand side, antigens in bacteria and that sort of thing. Important in the ability of cells to recognize each other, antigenic properties. So yeah, an antigen. Yeah, what is an antigen? I haven't read the exams yet, but yeah, what is an antigen? Could be a polysaccharide. But antigens are identity molecules on the outside of anything. So we'll see. Bacteria, viruses, or anything. What else have we got? Without your own antigens, you'd be in trouble. Connective tissues. So that underneath the outer layer of your skin, so you've got the epidermis on the outside, the dermis under that, and then you've got ligaments, tendons, all that sort of thing. There are polysaccharides that sort of hold it all together. Oh, moving a bit further down, we've got protective polysaccharides, heparin, produced by the, partly by the liver, but hence the hep bit, but it's secreted by most cells. It stops your blood clotting, heparin. And then gums and mucilages. Mucilage, I think, is one of those words that says exactly what sort of substance it is. Mucilage. Sticky, gooey on the outside of cells sometimes. The descriptions are good on the side. I'm not going to go through that. And then other food storages in certain plants that don't just store starch. Right, okay, that is carbohydrates done very quickly, but you know, enough really. It's just understanding carbon, hydrogen, oxygen, monosaccharide units, join them together to make disaccharides, polysaccharides. That's about it. All right, so we will stop it at that point.